Funaria introduction. It is a common terrestrial moss belonging to class Bryopsida of division Bryophyta having almost 117 species. About 13 species are found in Pakistan. The most common and worldwide distributed species is Funaria hygrometrica. It grows best in the presence of calcium, potassium, nitrogen and phosphorus. Habitat. The species of Funaria prefer to grow on damp soil, moist and shady rocks and other similar habitats. Some species grow on the decaying woods that found in moist shady places. Structure. External structure. The plant body of Funaria is gametophyte. Gametophytic phase consists of two growth stages, juvenile stage and leafy stage. A. Juvenile stage. This stage is represented by primary protonema. The protonema forms a brilliant green coating on the damp soil resembling algal growth. Many of the branches grow and spread over the moist soil. These are called chloronemal branches. The cells of these branches contain numerous chloroplasts. The other branches penetrate the soil. These are called rhizoidal branches. The cells of these branches lack chloroplasts. The protonema stage is only vegetative and transitory. B. Leafy stage. This stage is represented by gametophores. Gametophore is differentiated into stem and leaves. They bear sex organs at their apices. The leaves are small, ovate, sessile and green. The mature leaves have midribs but younger leaves are devoid of midribs. Internal structure. Stem. Cross-section of stem reveals a simple internal structure. The cells are arranged in three distinct zones: epidermis, cortex, central cylinder. A. Epidermis. It consists of a single surface layer of cells. The cells are small and contain chloroplasts in the younger portion of the stem. In the mature portion of the stem, epidermal cells become thick-walled and lack chloroplasts. The stomata are absent. Cortex. Inner to epidermis is cortex, which is of relatively greater thickness. The younger cells of cortex contain chloroplasts and lost when the stem matures. All the cells of younger stem are alike, but as the stem matures, the outer cells of the cortex become thick-walled. The cortex also shows isolated patches of small cells in the peripheral region representing the leaf traces. These traces do not join the central cylinder. C. Central cylinder. It is the innermost portion of cell composed of long, narrow and thin-walled colorless cells. These cells are dead cells and devoid of protoplasm. The central cylinder consists of two types of cells: leptoids, food-conducting cells, hydrates, water-conducting cells, leaf. A transverse section of leaf shows a well-defined central midrib and the one-cell thick lamina. The leaves also devoid of stomata. Life cycle or reproduction. Or vegetative reproduction. It takes place by following methods. Fragmentation. Under certain circumstances, protonema breaks into several fragments. Each detached fragment bearing buds may grow into a new plant. Gamete. Gamete are small bud-like structures developed from the terminal cells of protonemal branches. The cells of gamete are thin-walled and contain chloroplasts. They become detached from the parent thallus and germinate to form new protonema. The gamete may also develop on the stem and leaves of the gametophores under unfavorable conditions. On the returning of favorable conditions, these germinate into new gametophores. Bulbous or tubers. These are small resting buds developed on the rhizoids. These tubers remain dormant under unfavorable conditions and resume growth under suitable conditions and produce a new protonema. Or sexual reproduction. It is always of oogamous type. Species. Mostly homothallic and few are heterothallic sex organs. Antheridium male, Archegonium female. Alternation of generation. Regular and heteromorphic gametophytic phase. So it is the dominant plant phase. Represents haploid generation and develops sex organs. O. The gametophytic phase begins as haploid spores are formed. O. Sperms and egg are the last structures developed during this phase. O. Sex organs are developed on the leafy gametophores. In the monoecious or homothallic species, the antheridia are formed first and archegonia later. Antheridium. The mature antheridium is an elongated, club-shaped structure and has orange-colored body developed on multicellular stalk antheridiophore or male branch. The body of antheridium has an outer jacket layer of sterile cells called antheridial wall. This wall is protective in function and contains chloroplasts. The antheridial wall encloses a mass of small fertile cubicle cells called androcyte mother cells. Each androcyte mother cell divides to form two androcytes. The protoplast of each androcyte metamorphosed into a single biflagellate sperm. Each sperm is a minute slender and curved structure having a pair of whiplash flagella at its anterior end. The sperms do not release from antheridium until enough moisture is present to permit them to swim over it. Archegonium. It is flask-shaped structure. Develop on multicellular stalk archegoniaphore or female branch and consist of two parts. Venter. The basal swollen portion is called venter. It is surrounded by two-layered jacket of sterile cells called ventral wall. It encloses the venter cavity which is filled with two cells, upper and smaller ventral canal cell, lower and larger egg cell. Neck. The long and slender portion is called neck. It consists of 4 to 6 neck canal cells surrounded by single-layered jacket of sterile cells called neck wall. When the archegonium reaches maturity, the neck canal cells and the ventral canal cells degenerate to form mucilage. The mucilage imbibes water and swells. A neck canal is formed through which sperm enter into venter that containing egg. Fertilization. It takes place in the presence of water. Upon maturation, the roof of antheridial chamber disintegrates to expose antheridia which absorb water and swell. The upper end of antheridial wall ruptures to release sperms. The mucilage oozes out from archegonia and attract male sperms chemotatically. Many sperms move towards egg but only one of them gain entrance into the venter through neck canal and fuse with egg to form zygote. The act of fertilization ends the gametophytic generation and is the beginning of sporophytic generation. Sporophytic phase. The sporophyte is complex and partially dependent on gametophyte. It consists of foot, sater and capsule. The sporophytic phase represents diploid phase and begins as zygote is formed after the fusing of egg and sperm. The zygote increases in size and undergoes repeated cell divisions to form embryo. It almost completely fills up the venter of archegonium. The walls of the venter divide pericollinally and form a two-layered caliper around the young sporophyte. Structure of sporophyte. It consists of three distinct parts: foot, sater, capsule. Foot. It is the basal, bulbous, embedded portion of sporophyte composed of parenchymatous tissues. It is deeply inserted in the tissues of the leafy female branch. It attaches the sporophyte with gametophyte and absorbs food from gametophyte. Sater. It is a long, slender but tough, reddish-brown stalk-like structure and carries the capsule at its top. It raises the capsule more than an inch above the apex of leafy gametophores. The leafless sater is homologous to the stem of pteridophytes. Internally, it shows more tissue differentiation than liverworts. It is differentiated into a central strand of small thin wall cells surrounded by thick wall cortex and outer layer epidermis with a cuticle. Capsule. It is a highly organized, pear-shaped structure. It shows three distinct regions: A. Apophyses. B. Thecac. Operculum. A. Apophyses. It is slightly swollen and basal sterile region of the capsule. Its outer wall is epidermis that contains stomata. Beneath the epidermis, there is a photosynthetic region composed of spongy layer with air spaces and rich in chloroplasts. The center is occupied by a strand of thin wall cells that are conducting in nature. The presence of chlorenchyma cells with air spaces and stomata shows that apophyses is specialized for photosynthesis. B. Theca. It is the central part of capsule and is characterized by the presence of sterile central column of tissues called columella. It is surrounded by spore sac that contains spore mother cells. Outer to spore sac, there is a wide air space traversed by transverse strand called trabeculae which connect the capsule wall to the wall of spore sac. C. Operculum. It is conical and cap-like terminal portion of capsule. It is 4 to 5 layered in thickness. The lower two layers form annulus. The degeneration of annulus cells results in the loosening and dropping off of operculum. Below the operculum, there is a ring of tooth-like segments called sporophyte of Funaria peristem. The peristem teeth are hygroscopic in nature and respond to slight change in humidity. Sporogenesis. The sporogenous cell divides to form sporocytes. The sporocytes are the last cells of sporophytic phase. Sporocytes undergo meiosis to form haploid spores. Spore is the first cell of gametophytic phase. These spores later on germinate to form protonema that forms later gametogho.